Hello friends, uh, once again in 8th lecture we are starting with the metaphase 1 after viewing all the steps of prophase 1 in meiosis. Fine, if we very shortly revise all the steps of prophase, we have started with leptotene where only condensation of chromosome has happened in leptotene phase, that is prophase 1 first step leptotene. Now if you talk about meiosis step number 2 prophase 1, that is zygotine, there synaptonymal complex is formed and pairing of homologous chromosome is happened. Two bivalent chromosome have came across and they have joined together. In pectine of prophase 1, we have seen that recombination nodule have been formed which have converted two different bivalent chromosome into tetrad. So it is a pairing, complete pairing of two homologous different chromosomes. Crossing over is characteristic feature, key feature of the pectine, and at the point of the crossing over we can see it is having a recombination nodule where into the next phase, that is in pectine, recombination has to happen, right? So diplotin is known as Ch's metaformation, ready, where two sister chromatid of each of the chromosomes are getting separated apart from each other, started to separated apart, and into the last phase dikinesis we can see complete separation of the chromosome, but they are not detached from the place of chiasmata or recombination nodule. That is the only part left to get apart. Otherwise, all the chromosomes are getting separated from each other. It is completely fully, uh, we can say, condensed chromosomes, and the last phase dikinesis shows spindle formation, nucleolus and nuclear envelope disappearance. So that is a story of prophase 1, and we are supposed to go for the other phase, that is metaphase 1. Now if I talk about metaphase, metaphase in mitosis and metaphase in meiosis have large difference with respect to their arrangement of the chromosomes. Now, if I talk about the metaphase of mitosis, we know that when it used to be a cell, chromosomes are arranged into the center. For example, this is one maternal, this is the copy paternal, the other here, and its complementary chromosome is here. So homologous chromosome are arranged into single plane, equatorial plane, only a single plane, ready, because it is not having pair of homologous chromosomes. It is absent here. So they are arranged into a single plane. single equatorial plate. Now, due to that, their spindle fibers are arranged such a way that one single chromosome, for example, if it is a chromosome, at both the end it is joined with the spindle fiber. Ready? So spindle fiber are joining at both the end, from south pole and from north pole. Two spindle fibers are joined with both the side from centromere. So at the centromere, two spindle fibers are attached. Now if I talk about meiosis, that is meiosis 1, what we are about to look at, we have pair of chromosome. So this one is arranged in this pattern. I hope you are very clearly getting this point. Ready? And this is the synapses. This is the synapses where they are joined together. That's where we have ended our prophase 1. And this is the why one chromosome, that is one bivalent chromosome, is attached to either south pole side spindle fiber or north pole side spindle fiber respectively. Ready? For example, this is south pole spindle. So that was north pole spindle. And they are arranged in either of the chromosome. So here if I talk about the arrangement, they have pair of homologous chromosome, and that is why they are attached into single spindle fibers. One chromosome and the other chromosome. So that way they are attached to single spindle fiber at each side of them. So this is how the arrangement of two or double layer of metaphase plate or equatorial plate is to be considered. It is double layer of equatorial plate or metaphase plate. So it has got double layered metaphase plate. Why? Because of pairing of homologous chromosomes. So this is the reason why it is having a double layered metaphase plate. I hope it is very clear to your mind. Ready? So let us consider that matter here. So in metaphase 1, what you can see is it is having a pair of chromosomes and it has to arranged into the metaphase plate like this. So one chromosome towards south pole and the other chromosome is attached toward north pole spindle fibers, what we have just recently discussed. Ready? So spindle fiber formation is completed, right? It is completed here into this phase. Now chromosomes align on the equatorial plate, that is these. Now microtubules from the spindle attach on the pair of homologous chromosome. Entire pair is connected with one side of spindle fiber. So this is the speciality of the metaphase 1. So here one important point left is two layers of metaphase plates. Ready? Mitotic plate or equatorial plate, what happened is at the two step or two layer. Fine, I hope you are very clear about this now. Fine, so please take a screenshot, then we can go ahead for the further step. Now comes the anaphase, where chromosomes are getting apart from each other. They are moving far away from each other. Fine, let's compare and discuss the anaphase. Similarly, what we have discussed here about the metaphase. Anaphase. If I talk about mitotic anaphase, we have cell, we have the chromosomes, and those chromosomes are actually joined at the both the sides. So here thing is that during anaphase, condensation or stretching of the spindle fibers toward their respective pole, contraction of spindle fibers are used to happen during anaphase. So what happens is that during mitosis in anaphase, both the chromatids are getting separated and moved toward their respective polar regions. So into anaphase of mitosis, chromosomes are separated or broken down from centromere, right? So two sister chromatids will be separated. So that's what happened during anaphase of mitosis. But in meiosis, what happens is that we know that we have pair of the chromosome, not a single chromosome. It is arranged into the pair. For example, if I arrange like these, then condensation definitely has to happen, right? It is condensing, or the contraction definitely is going to happen at their respective polar region of the spindle fiber. But entire chromosome is moving towards their polar region. That is what the difference. So there is no separation or broken down from the region of the centromere of any of this chromosome. So entire chromosome, entire bivalent chromosome will move towards their polar region. So that's the thing which is happening into the anaphase. So anaphase shows only one event, that the homologous chromosome separates, but two sister chromatids remain associated with each other at the part of the centromere. It is not getting detaching. So this is the speciality what you are about to remember, and that's the difference between anaphase of meiosis and mitosis. Ready? So I hope you are clear with the idea of difference in the metaphase and anaphase of respective mitosis and meiosis. So please take this screenshot so that you can have a better note. And last one, the telophase. Now during telophase, two nucleus has to formed. Ready? Two nucleus has to formed. So whatsoever chromosomes which are getting separated from each other during anaphase are actually getting accumulated towards their north pole and south pole of respective sides, and they have actually developed nuclear envelope around them. So this is anaphase, if you see, right? This is anaphase, where contraction of mitotic spindle is happening, and that is why the chromosomes are moving either from south pole or from north pole. And after they are reaching to their respective pole, this is a nuclear envelope which is actually formed around them, and centromere is getting nearby to the nuclear envelope. So two nucleus are formed, and later on cytoplasm also divided. Most important thing is that here you can produce you can produce two cells, two daughter cells, by consecutive cytokinesis as well. So karyokinesis is done and cytokinesis also done, and two daughter nucleus is produced there. But condensation of the chromosome remain as it is. So chromosomes are not converted back into the form of the chromatid. Why? Because the second division is still left. It is just the end of meiosis 1. One more division is still left. That is why, especially after division number 1, that is meiosis 1, chromosomes are remaining into the condensed form and they are not actually decondensed. They are not converted into the chromatin back till the second division is finished. So that's the specialty you are supposed to remember. Right? So telophase, if I talk about, two nuclear membrane and nucleolus reappears. So you can find two cells. So two haploid daughter nuclei are formed. The thing is that haploid daughter nuclei are formed. Why? Because at the time of telophase 1, either paternal, mind it very well, or maternal, both the chromosomes which are actually arranged into two planes of the mitotic plate, they are getting separated. So one side it is paternal, one side it is maternal chromosome. So both the polar region are getting one single type of chromosome, either maternal or paternal, and that is a reason why maternal plus paternal mixture is separated. Diploid condition is no more, though number of chromosome is doubled, but both were either paternal or both are either maternal. In that sense, we can say that it is the haploid nucleus. So number of chromosome actually are, though it is double, it is either maternal or paternal, and in that condition we are saying it as haploid daughter nucleus. So it is reductional division in that sense, right? So after these, cytokinesis may or may not occur after short interkinase. Interkinase is a phase where it is a very small break between meiosis 1 and meiosis 2. It is followed by meiosis 2, and the short stage between two meiotic division is interkinesis. So DNA replication does not occur in the second phase. Only once the replication is done. Second time, at the time of the division, DNA replication does not occur. So that's what also to be remembered. So that's the story of telophase. Ready? So two daughter cells are formed by the process of telophase 1. Fine. Take the screenshot and then we are moving ahead. Now, story of meiosis 2, where two cells are formed by each two daughter cells. So total number of cells which are produced are 4. So till now we have gone through meiosis 1, where we are producing two daughter nuclei which are haploid, right? So at the end of this lecture, with two daughter nuclei. We are concluding that Meiosis One is ended with the two daughter cells which are haploid, so two haploid daughter cells, and that is why we are concluding as reductional division. Now what happens is that after that means after the interkinesis, we can see that meiosis 2 has to happen, where both of these 1 and 2 cells are dividing to produce two more cells of each. So total 4 cells will be produced into the meiosis 2. So meiosis 2 will be having a similar 4 phases, that is prophase, metaphase, anaphase and telophase, and that will be that will be discussed into the next lecture. So till then, I'll see you later on. Take care and have a nice time.